currently there's a lot of mouse models of different diseases that exist that are used to test different hypotheses on how the human disease might work. So for example, there's models of multiple sclerosis in mice, there's models of diabetes in mice, but nobody really knows how exact those mouse models mimic the human model. And obviously we don't make human models of disease, right? So the models that we study are actually the people that have the disease. You can't go back and clone them. But what you can do now with IPS is that you can take, say, a skin cell from a person that has multiple sclerosis and reprogram it to an IPS-like state and kind of study how it develops into a neural cell and see, for example, if there's some developmental block. And so you're actually using a cell that comes from the patient, so it should be more accurate in terms of the disease that you're trying to study. Or at least that's the idea. The other idea is to use IPS cells from patients to test drugs. So if you were to make an IPS cell for a certain disease and differentiate it into a specific cell type, you could just have all these different cell types in culture and throw whatever chemical or drug you want on it and look at whether that drug is toxic, whether it really mimics something that happens in the disease. You can screen drugs before having to go into clinic and testing them in patients. So they're kind of being used as a screening technology now too. So they're also been used to cure certain diseases in mice. So this paper came out in 2007 where a mouse model of sickle cell anemia was cured of sickle cell disease using iPS cells. And so this is one of the early papers that have shown that iPS cells can actually be used to treat diseases. And so this is kind of a very complicated experiment, but what they did was they took the fibroblasts from a sickle cell mouse, they reprogrammed them into iPS cells, and here what they're showing you is they call it H-beta S, so this is human hemoglobin of the sickle cell type. Okay, so sickle cell anemia is caused by the mutation in the hemoglobin protein that causes the cells to be misshapen, and they can't carry as much oxygen, and they also could clog arteries. So it's a simple point mutation. So if you take those iPS cells, and then you can correct the sickle cell point mutation using genetic engineering, and so you have an iPS cell that now expresses the normal hemoglobin, that's hemoglobin A here. And then differentiate those cells into more hematopoietic-like cells, and then put those back into a mouse that has sickle cell disease. What they found was that the animals actually improved. So they were able to see that in the animals that got the iPS cells with the corrected sickle cell gene, that they expressed hemoglobin A, which is the normal hemoglobin that's shown by this peak, as compared to the untreated sickle cell control mouse, and that these mice actually did better. They were actually healthier. So this is proof of principle that iPS cells generated from a tissue like skin can be redifferentiated back, or de-differentiated back into a pluripotent-like state, and then redifferentiated into the cell you want, and actually have some beneficial effects. So I'm sure you'll see in the future a lot more diseases that are kind of being treated with this kind of technology. Okay, so that's what I have today. So there's different ways to reprogram somatic cells, okay, not just iPS. And this is an example of how developmental biology concepts can explain how somatic cell reprogramming occurs and how you can use developmental biology concepts for certain applications in terms of medicine, drug development, tissue engineering, what have you. Okay, so if you have any questions,